So I thought I'd just start by giving a kind of a little bit of picture of what I mean by the dimensionality and dimensionality reduction in high-dimensional data sets. So I thought I'd use a kind of an intuitive example to lead into this. The example I've chosen is reading handwritten digits, so numerical digits written by hand. Now we think of this, recognizing a numerical digit is, we don't consider it a difficult problem because we do it so easily and naturally. But if you try to teach a machine to do it, then you kind of see you kind of see where the high-dimensional issues come in. So here is an example. Can you see my mouse, by the way? Yeah, you can. So here's an example of an image of a handwritten digit, happens to be a number 4, and you can see the pixels here. So in order to specify one of these images, I need to give the grayscale value for each one of these pixels. And so each one of these images sits in a space which has a dimensionality equal to the number of pixels here. So the space of possible images is very high dimensional, but the space of handwritten digits is actually is kind of low dimensional because there is a structure and a pattern to the images, such that you can represent them here. I'm showing a PCA plot of the handwritten digits, and you can actually see the main structure in the data in just the two-dimensional surface of your screen there. And this is something that's sort of generally true of biological data, even though they reside in very high-dimensional spaces, because of biological systems are sort of highly constrained, and with lots of structure. Even though they sit in a high-dimensional, they're embedded in a very high-dimensional space. The structure in the data resides in a low-dimensional sort of manifold, and this is why, for example, principal component analysis is useful in biology. Here's a nice example looking at principal component analysis of gene expression across many different tissues in a human, and you can see in two dimensions you can pick out lots of most of the structure in this data. So this is going to be like this picture sort of summarizes what I'm going to be talking about today. We're going to take a data in the form of a very large matrix. So here is some gene expression data, for example, and across the columns we have lots and lots of genes, 17,000 in this example, and down the rows we have samples. And so we have this very large data matrix, and each data point is embedded in a very high dimensional space. We're going to actually find that the main structure in the data resides in a much lower dimensional space. So this is, in terms of data matrix, this is what what this looks like. We're going to take a data matrix and we're going to decompose it into two parts. One is a highly structured, sort of low dimensional, in matrix language, a low rank object matrix. And then the other part is a very high-dimensional, unstructured, what you might think of as noise. So we're going to try and separate the low-dimensional part, the low-dimensional structured part of the data from the high-dimensional noise part of the data. So I'm using this term here, low rank. So this is a low-rank matrix. This is a high-rank matrix. If I'm going to use this term, I should sort of define it a bit more carefully. But the intuition is that it's the dimensionality of the matrix. So the strict definition of the rank of a matrix is it's the number of sort of linearly independent rows. That is the number of rows of your matrix that can't be expressed as just a linear combination of the other rows. So another way to think of that is in terms of the singular value decomposition. So here D is our data matrix, and we can represent that as a product of three matrices. Hell, I've lost the sound. Can you hear me?
Oh, we can hear you. Yeah, very well.
I'm sorry. Sorry about that. So the data matrix is decomposed into three parts. U is a unitary matrix, D is a diagonal, and V is another unitary matrix. So here is like written down in a simple example, a very small, say 3x5 data matrix, and you can express this as the product of U, D, and V. So U is an orthogonal matrix, and V is also an orthogonal matrix, and they form essentially an orthogonal basis for the matrix. And the middle part, D, is what's called the singular values down the diagonal. So this is a kind of a scaling parameters. Now the number of non-zero singular values is the rank of the matrix. Okay, so here the matrix has rank 4 if all of these values are non-zero. But what you often find with data that has a embedded in a high dimensional space, but it has intrinsically a low dimension, what you find is that some of these singular values are very much smaller than the others. And what that means is if you truncate them, so you write a version of the singular value decomposition which is truncated on the higher order singular values, so it starts to look like this. Here we've chopped off the last one. What you find is you get a low rank version of the matrix which is almost the same. It's a very good, it's a good approximation. So you can get a version of the data matrix which has a much lower rank, often like dramatically lower rank, but is actually still quite a good approximation for the original data matrix. And this is going to be one candidate for this extracting the structural part of the data and separating it from the random, the noisy high dimensional part of the data. So why am I talking about this? It's starting to become more important, these issues, especially in with the development of big data. What we're finding is that the scalability of statistical approaches is becoming extremely important, because as data becomes extremely large, traditional statistical methods, they're prohibitively slow and computationally expensive. So it's a practical issue that we can't use them. So finding new statistical approaches which are able to scale to the extremely large data sets, such as we're going to see in links, is becoming very important. There's other practical concerns with very high dimensional data, which is data handling. Just storing the data, transferring and sharing the data becomes very cumbersome as the data becomes very large. And dimensionality reduction is one approach that can alleviate these problems. You can, by reducing the data down to its essential signal, you get a much smaller representation of the data, which is easier to handle. It makes statistical analysis of the data very much faster, sort of exponentially faster. And I'm also going to show you get the added benefit that it actually increases the accuracy of statistical analysis. We're going to get better higher statistical power, as well as solving these other practical issues. So there are many ways to approach dimensionality reduction, but for concreteness, I'm going to describe one, and I'm going to show you examples of this in my talk. And here we're coming back to the singular value decomposition. So we're going to take our matrix and we're going to think of it in its singular value decomposed form, so the product of those three matrices. And what we're going to do is we're going to find a low rank approximation to the data. So we're going to find the matrices which minimize the difference between the low rank matrix and the original data. And we're going to do something else as well. In addition to that, we're going to use regularization penalties. So this is something that's often important in high-dimensional data, and it's a process that helps generalize the results of the statistical analysis. It makes it generalizable and not essentially we're avoiding overfitting data. I can describe the regularization process in a bit more detail. Here's a nice illustration here where we've got some data points, the little circles here, and we're trying to fit a function to this data. So we're finding the function f, which minimizes the difference between its value and the data value. So one way to do it without regularization is to find the function that sort of wiggles around, matching all the noisy fluctuations, and this way you get a perfect fit. But when you bring in some more data that has not been used to train this function, you're going to find it doesn't work so well because you've fitted the noise. So what a regularization scheme tries to do is it says, yes, I want you to match my data as close as you can, but also I want you to be smooth in some sense. And you can do that by including a penalty term in your loss function. So here's what it looks like. You know, okay, yeah, I'll fit my data as best you can, but also be smooth. So don't try to abuse the noise to get a good, a good least squared value. So I'll be doing that in my examples here. Okay, so now I can start to show you some examples. The first example I have is from L1000 gene expression data of drug perturbations. So just as a kind of a trial, I've taken 730 samples from L1000 from the LGP5 set, all in the MCF7 cell line, and I've taken a couple of the replicate sets. So in this smallish subset of the L1000, we have 59 unique drugs. They occur in replicates. I'm not counting DMSO in my in this example here. So, before I carry on, I should note that dimensionality reduction is sort of already implicitly used in the L1000, in that L1000 takes 1,000 landmark genes as being representative as the full 20,000 or so genes in the genome. But what I'm going to show you is, we can take alternative routes to this dimensionality reduction and take it even further with a number of benefits. So here is my the original L1000. I've missed off a 0 here. This should be L1000 data. So here are the landmark genes all across the columns, and my 700 samples down the rows. And this is what the original data looks like. And then when I go and solve this singular value decomposition, and I truncate and I regularize, I get this version of the data. I get what looks like someone in the lab called it a cartoon version of the data, which is sort of a nice description, because you're sort of missing some of the detail here, but we're trying to distill this data set into a simple low-dimensional form here. And you can kind of, I included this picture because you can kind of see it visually. So we've actually done this decomposition. Here's the original data. Here's my low-dimensional part. So this is the part that's supposed to contain the structure in the data, or the essence of the structure in the data. And then if you and we also have the noise part here. So if you add these two matrices together, you get the original data back again. So now I have two representations. I have the original data, and I have a dimensionally reduced version of the data. So what I'm going to do is I I'm going to train a classifier to try to predict what is the drug that, given a gene expression profile for the landmark genes, what is the drug that made this perturbation. So and I'm going to assess the quality of this classifier using cross-validation. I'm going to use tenfold cross-validation, and then we're going to compare the accuracy of the classifiers. So here on the left, so if I try to guess the drug in an uninformed way, I would expect to get the right drug about 2% of the time. With the classifier on the original data, I get the right drug about 35% of the time. But with the dimensionally reduced data set, I'm getting it right closer to 50% of the time. So it's a modest increase in accuracy, but it's a real increase. If you test for the significance of the difference, it's got a significant p-value, the order of 10 to the minus 4. So it's a modest, but it's a real difference in the accuracy. So what this means is, well, there's other ways of sort of observing this difference in accuracy as well. We can look at the accuracy as a function of rejection rate. So what this means is, if we set a threshold below which, if the probability of belonging to a particular class is too small, we reject that data point as being unclassifiable. We can look at the accuracy as a function of that rejection rate. So you can see clearly the reduced dimension data set is given a much higher test accuracy. So just to be clear, this is an estimate of the test error. So we're testing on gene expression profiles that weren't used to train the classifier here.
Neil, can I interrupt you just for a second?
Sure.
So do you perform any feature selection in your classifier construction process?
No. So I've taken the other approach where, so I'll just go back here. So you can see I'm actually using this data set. I've not reduced the number of features, but I've reduced the number of kind of modes of variation. Is another way of saying it. So all of the landmark genes can contribute to one of these modes of variation, but there are only a small number of these. In fact, there are 30 modes of variation. So I'm not selecting a subset of landmark genes exactly. I'm selecting like representative patterns of gene expression.
Right, but I mean, if it just occurs to me that maybe you could think of actually using this principal components as reduced dimensionality, right? I mean, not really, that's what it is, I guess, as features, right? So it is kind of almost like feature extraction process, right? You extract,
yes.
Right?
Yeah.
Whereas for the other one, you don't do that, so you use the complete kind of set of features that may be noisy. So I'm just trying to see, where exactly the difference comes from.
Right. So yeah, I mean, the difference is kind of, I tried to illustrate the difference here in that I've tried to get like a cleaned up version of the data file. So the kind of the low rank part of the data. So I still have exactly the same number of features. I have 1,000 landmark genes, and I still have 1,000 landmark genes here, but I've got fewer kinds of modes of variation. So yeah. Okay. So
I understand. I think I was wrong. Yeah. So you don't actually use the kind of labels, right? You don't perform supervised learning for extraction of features. You do unsupervised feature kind of creation
yes,
by the I got you. All right. Sorry.
That's right. Yeah, I'm the supervised part comes in the in this part where I'm trying to compare compare the accuracy of classifiers built upon the two data sets. Yeah. So this is unsupervised, and I'm using supervised learning to try to assess the quality of statistical analyses on the two data sets. So we get an improved accuracy with this reduced dimensional data. We get the added benefit that it's actually a lot quicker. It takes about half the time. It took 71 seconds to train the classifier in the original data, 29 on the reduced data set. So there's about a factor of 2 difference here. But as the size of the data sets increases, this difference sort of increases exponentially. So on big data sets, this will actually be orders of magnitude difference. I can show you an example of that later. Here's another example. I'll do this one a bit more quickly, but it's because I don't want to be repetitive. It's the same kind of idea. I've taken tumor samples from TCGA. I've got gene expression and DNA methylation for these tumor samples. And amongst these 375 samples, I have representatives of all these 5 breast cancer subtypes. So, and I'm using the DNA methylation data, which is shown here in the top left. I have the DNA methylation data, my 375 samples here, and DNA methylation probes along the columns. And I do the same thing. I make a reduced dimension version of the data. And then I build a classifier to see if I can find subtype-specific patterns of DNA methylation. So I'm getting the same kind of results here. The original data, I can predict the subtype of breast cancer from the DNA methylation profile with about 51% accuracy, and this is improved to about 59 on the data the reduced dimension data set. And again, this is a small, but it's a real difference. There's a very small p-value on a Mann-Whitney test of this. And if you look, is a slightly we get a slightly bigger difference in the time, the computation time here. There's about an order of magnitude difference. Calculating on the dimensional reduced data set is about 10 times faster than on the full data. So we see, so making the point that it's this sort of cartoon version, the low dimensional data set is giving us more accurate classifier. If we come into the confusion matrix, we can try and sort of pick apart what exactly this data set is doing better. So down the rows here we have the actual class of a test sample, and along the columns we have the predicted class. So what we would ideally like is all these to be sitting on this diagonal here. And what we find is that with the reduced dimension, dimensionally reduced data set is on the right here. What we find is we're able to pick out the normal-like samples with the reduced data set, but we're not able to do that on the original data. So I think what he's showing us is this reducing the dimensions is actually cleaning up the signatures, and it's actually helping us to get a clearer picture of what the data is telling us. It's a little bit unintuitive, really, because we're kind of throwing away something when we do this. We're taking all these measurements and we're reducing them down to sort of 30 representative profiles. So we're actually throwing something away, but in throwing something away, we're actually getting greater accuracy in our statistical analysis, with the added benefit that it's actually a lot faster as well. I'll skip past this because this is going to get very repetitive. Here's another example where I try from gene expression, try to predict whether a patient will experience a new tumor event, either a metastasis or a new primary tumor, for example. And I can do this on original gene expression data and reduced gene expression data. And we see the same sort of thing. We get a gain in accuracy. And here we've got a much more dramatic difference in computational time. We've got 3 orders of magnitude here. So it's actually 1,000 times faster while being more accurate. Okay, back to links data. This is a slightly different example, and I'm using a slightly different approach to dimensionality reduction. So the data set I'm using is from science signaling, and we're looking at profiles of post-translational modifications in 31 cell lines based on mass spectrometry. So we have about 17,000 post-translational modifications that we're looking at here, and in 31 cell lines. Now, because we're using mass spectrometry in this data, we're not always able to pick out the peak, so we're getting some quite a lot of missing values here. So we're modifying the dimensionality reduction technique to only use the values that we know, which are represented here as omega. So this is our original data, and this Z is a low rank approximation to the original data, which is constrained to have regularized to have a low rank. So here's what it looks like. Here is the original data. I've just selected 100 post-translational modifications. There's actually 17,000, but I've just picked out a few to resolve the matrix. And here's the reduced rank version. So here we've got this low rank version of the data, but we've got the added benefit that we're actually able to fill in the missing values here. We're sort of taking it, essentially we're taking advantages of patterns and structure that are present, that are in the present values here, to fill in the missing values. So the next thing we did with this data is we built a graphical model. So we're looking at a network of post-translational modifications based on this data, and we get one network for the original data and one network for the dimensionally reduced data. And then we want to assess the quality of these two networks. And one way to do that is to evaluate the degree to which prior knowledge of pathways are present in these networks. So we selected all KEGG pathways, and we look for we count how many KEGG pathways are represented to a statistically significant degree in each of these networks. So that I've sort of written them like a bar graph, because the idea is that the height of this list should be representative of the quality of the network. So on the left here, I have the original data. We build in, we get 8 KEGG pathways represented. This tall curve here is from the second data set on the reduced dimensionality data set, and here we have 43 KEGG pathways. So this is kind of showing that with reduced dimensionality, we're actually able to recover more known pathways. The idea isn't just to reproduce KEGG, but the idea is that the degree to which we are consistent with known prior knowledge is going to be a measure of the quality of these networks. So what I've shown you so far is, if you take the data, you reduce its dimensionality, you can very often get increased accuracy if you're a statistical analysis, while also getting while also being much faster. Okay, but then the question becomes, how much do I reduce the data? Like Einstein said, everything should be made as simple as possible, but no simpler. So, because in reducing the dimensionality of the data set, we're throwing some information away. It's So if we reduce it too much, we're going to start throwing away very informative part of the data. So the next question is, what is the intrinsic dimensionality of the data? There are many methods to address this question. They can be broadly classified into three. Local methods use sort of local information, so they're looking at the nearest neighbors of each sample to try to estimate the topological dimension of the manifold, of the data's manifold embedded in the very high dimensional space. Global methods take the data set as a whole. Another broad class is the fractal methods. So these are the ones like box counting. So this is the famous example of trying to estimate the dimensionality of the coastline of Great Britain. You cover it with boxes, and you look how many boxes does it take to cover the coastline, and you count that number of boxes as a function of the size of each box, and in the scaling gives you the fractal dimension. This would be it would be very nice to apply this on gene expression data, but unfortunately there's a relationship between the dimensionality that you can capture with a given sample size, and it's logarithmic. So this is a kind of a problem. You need extremely large sample sizes. However, with the links data, we might actually get to the point where we're able to use this. In the meantime, we have been developing a new approach to assessing the intrinsic dimensionality of links data in particular. And we're similarly to the fractal-based methods, we're going to use scaling, but we're going to use scaling with sample size. So I've made a little illustration, a very simple illustration to explain what I mean by that. So here are three cases. We'll work in 3 dimensions because it's easy to visualize, but we're actually going to apply this in much higher dimensional spaces. So you can think of this as a 3-dimensional gene expression space, and each one of these points representing a microarray. Now, if there are no constraints on this system, the points are just going to fill the space, and they're going to spread out in every possible direction. If we add one linear constraint, what will happen is that the points will actually lie to a large degree on a plane, a linear surface in this space. And if we had 2 linear constraints, we'd actually fall on even a lower dimensional manifold of a straight line. So now we're going to start this. I'm going to try to explain this this idea of sample size scaling to estimate dimensionality. Let's start with a single sample. Here's the number of samples on the horizontal, and here is the dimensions of the space that we need to capture the variation here. So we've got one sample, and we can capture it in a 0-dimensional space, just a point. 2 samples, they lie on a line. There's a straight line that always connects 2 samples, 2 points. So with a 1-dimensional subspace, we can capture the variation in our data. Now let's skip ahead to 6 samples. Now if these 6 samples are spreading out in every possible direction, we're still going to need all the principal components to capture the variation. But if they're actually lying on a lower dimensional manifold, we're only going to need a few. We're going to need fewer principal components, and you can sort of extend this further. Okay, so the idea is that if the data is actually falling on a lower dimensional manifold, the curve of the number of dimensions required to capture the variation of the data will actually level off or scale with a slope of less than 1. So here we've applied this to the breast cancer samples. We have the 375 breast cancer samples, and they're divided into, we've split them up into their 5 different subtypes, and we're looking at how the dimensionality of these data scale with the sample size. So here's the legend indicating the subtype. And what we can see is there's a different scaling for different subtypes. And we can actually extrapolate the curve to estimate what kind of sample size we would need in order to completely characterize each breast cancer subtype. And we can do this in the transcriptome space, the methylome space, and the proteome space. So we can actually get a subtype-specific representation of the dimensionality of these data. Another thing that we notice here is that this seems to be an apparent, just an interesting observation, is that there seems to be an apparent relationship between the dimensionality and the different levels, such that a higher dimensionality, if a subtype of breast cancer has a higher dimensionality in the transcriptome, it tends to have a higher dimensionality in the methylome and the proteome, which I suppose is perhaps not too surprising, but it's I think it's interesting to observe it in this way. So this kind of a process could be used in links data sets to estimate when we're done, when have we got enough information that we've completely characterized the space of perturbations, or we've characterized it to the degree that we need for analysis. So, in summary, what I've tried to show is that dimensionality reduction is something that's going to be very useful for links data. It helps alleviate some practical problems of data storage and data handling, sharing, transfer, for example. And it can also lead to some significant increases in the accuracy and statistical power of analysis of this data. It makes for some very scalable statistical analysis that can actually, you can actually run the analysis in a reasonable amount of time. And I've also, it's in the examples I've shown with the L1000 data, the implication is that with the reduced dimensionality data sets, we actually get a cleaner signature for the perturbations, and this is perhaps the mechanism that we're getting this increase in statistical power. So thank you very much for your time, everyone. I'm happy to take any questions.